Welcome to Biology Made Easy. In this lesson, we are explaining DNA replication. We'll do part 1 because in this lesson, we'll explain the action of DNA polymerase and continuous replication. In part 2, we will still discuss DNA polymerase, DNA primase, RNA polymerase, primer, ligand strand, Okazaki fragments, discontinuous replication. Welcome to part 1. Well, in DNA replication, we have one DNA molecule duplicating into two DNA molecules. DNA is supercoiled and the enzyme topoisomerase relaxes the supercoiled DNA and exposes the double helix to be copied. So, after topoisomerase have disentangled it, the enzyme helicase breaks the bonds between the nitrogenous bases, so exposes the bases to be copied. The enzyme DNA polymerase copies one of the strands continuously, and we want to find out which strand. DNA polymerase is said to move in the 5 to 3 end. What does it mean? This DNA molecule, this is the 5 prime. We can trace the 5 prime, and the end of the 5 prime is the 3 prime. Well, take note that the old DNA strand is the black one, and the new DNA strand is the red. So the 5 prime ends up in the 3 prime, and DNA polymerase moves in 5 to 3 end of the enzyme. Now, let's use this to explain. This is the 5 to 3 strand. Does it mean that the DNA polymerase moves on the 5 to 3 strand? No. The DNA polymerase is moving to fit in nucleotides in 5 to 3 end. So, let's use this to explain what the 5 to 3 end means. This is a DNA molecule. This is one strand. This is the other strand. This strand starts from the 3 prime and ends on 5 prime. What that means is that the 3 prime is the 3 carbon. When you say 3 to 5, we are talking about carbons. So you have carbon 1, 2, 3, 4, 5. This strand starts on carbon 3. That is why it's 3 prime. And it ends on carbon 5. So the 5 prime. 3, 5, 3, 5, 3, 5. That is this strand. This strand, we have carbon 1, 2, 3, 4, 5, starts on carbon 5. So you have carbon 5, carbon 3, carbon 5, carbon 3, carbon 5, carbon 3, and so you have 5 prime and it ends on 3 prime. That is how the DNA molecule is. And the DNA polymerase is moving on 5 to 3 prime, not on this strand. Not on this strand. The DNA polymerase is moving to form another strand that is from 5 to 3. So the DNA polymerase will have to copy, and in actual fact, copy this. So the DNA polymerase is forming another strand, and the strand that the DNA polymerase is forming is 5 to 3. It's forming a complementary strand for this strand. So DNA polymerase moves to fit in nucleotides to form a new strand that moves in 5 to 3 prime. So the DNA polymerase does not move on the old strand, which is 5 to 3. That is why in this one you have DNA polymerase moving in 5 to 3 and forming this new strand that has 5 and the end will be 3 here. And DNA polymerase moves continuously, fitting in 5, 3, what is 5, 3, 5, 3, 5, 3, 5, 3. So for this old strand, which is the black one here, you see it starts from 3 prime. DNA polymerase fits in continuously 5 to 3 end and is continuous. It forms a complementary strand starting from 5 prime to 3 prime to the old one that starts from 3 prime to 5 prime. So this is what continuous replication is, and this strand is said to be the leading strand. In our next lesson, we will look at this strand, which is the discontinuous replication. If this lesson has been useful, please give us a thumb and please subscribe. Thank you.